Pressing, I know.
But that's, I mean, you can see that as depressing, but I see that as a way that in silico screening, there are a lot of opportunities in silico screening, and people are discovering them as they can. It's not just a defined tool that you use in a set way. There are areas for it to grow.
So just to further maybe explain, so I'm not a believer, I think you figured it out by now, I'm not a big believer in structure-based docking. People use it, but not very often. So my group use it maybe in 5% of the time. So 95% of the time we don't even apply it at all. Bear in mind that the projects that we're getting are many times phenotypic projects, so we don't even know what is the target.
Okay.
And the other projects that we're getting are against novel proteins, transcription factors, stuff which we don't even have a clue how the structure of the protein look like. So these are the kind of project that we're getting. So I'm not saying that we don't get kinase projects and more well-established target families, but even with those kind of projects, we're being challenged to find novel modes of inhibition, for example, allosteric inhibitors, that if you use docking approaches that you need to predefine what is the binding site, and again, docking or structure-based approaches don't work very well. So I can tell you what other approaches we do apply, but ligand-based approaches or structure-based approaches are not always applicable to lead discovery.
Meir, then I might start off after I talk to you about you personally, your title and what you do day to day. Then that might be the first question to talk about different approaches to screening molecules for activity in silico. And then if you could kind of frame that as there's this ligand approach and structure approach, and clarify that a little bit, and then we'll take the questions from there. Does that sound okay?
Yeah. So there are structure approaches which are primarily docking, that you try to fit a compound into a binding site. And there are experts in the field that like to build homology models and use more detailed, I would say, molecular simulations to do that. There's another niche that uses another approach that uses the molecular structure as the probe, and the idea is to run a chemical similarity search, so look into compounds that look the same. So the idea is that compounds that have similar chemistry will have similar biology. And there's a lot of activity around the field of developing descriptors. There are 2D descriptors that rely on the topology of the compound or the scaffold of the compound. There are 3D descriptors that think what is the pharmacophore, for example, and what is the shape of the compound. Each method has its advantages and disadvantages, which I'm happy to discuss. There are also biological approaches, which works in my mind the best. So what you do is that you don't worry how the protein structure look like, you don't worry how the chemical structure look like. What you do, you actually use the bioprint of the compound as your description. So what we have, for example, is a screening collection of, say, 2 million compounds, and they went through hundreds of assays, right? So you have a very big matrix of the biology or the bioprint of the compound. So what we found that works the best, if you have enough biology data, is using this bioprint and saying that compound that has similar bioprint, like against target X, is also so compounds that have similar bioprints tend to behave similarly. So if you want to drug a certain target, just find and you have a compound that inhibits this target, find the other compounds that have the same bioprint. This works the best, in my opinion, in terms of enrichment. This also brings back a lot of novel chemotypes. So that means novel scaffolds, which is very critical to drug discovery. It's critical from the patent space. So if you have, for example, Merck published a new compound, so what you want to do is bioprint their compound and find other chemotypes, and that hit this target that will take you to a new IP space. Or if someone published a natural product that has kills cancer cells, so very interesting to you, but it's not very amenable to chemistry. You could use the bioprint, and it will take you to a synthetic space that you could maybe work with. So I'm a big fan of this kind of approaches. You could apply phylochemical and phylogenetic approaches. So that means that if you have target X and you want to find new inhibitors for this target, you could ask yourself, what are the phylogenetic neighbors of this target? Which compounds have similar, proteins have similar sequence or bioinformatically are similar to the target that you're interested to inhibit. So maybe you're familiar with the Sujan kinome tree, or any way to describe the similarity between proteins based on bioinformatics principles. So if you think about like evolution trees, or in terms of the sequence, the amino acid sequence, how similar are they? And what you want to do is identify other proteins that show high degree of homology or sequence similarity to your target, and then you test those the compounds that modulate the neighbors or the bioinformatically similar proteins. So this is the phylogenetic approach. You could apply phylochemical approach, that you could build your own trees or classification trees of protein families based on the similarity of the ligands that they're binding, and then design sets based on that. There's an immense amount of knowledge in biologically annotated databases. So we invested a lot of money in curating our in-house data and buying external data and so on. So just by even going to the literature, which is well curated in our hands, and just asking, what do you actually know about this protein family or about this protein, you're sometimes surprised with what we get back. And I believe much more in actually data than virtual screening approaches. Many times just data itself is available, and I would argue that about half of the data that we generate is redundant. If we could actually utilize it and leverage it effectively, we could get much more than today with what we do. So my mindset is less about physics-based approaches and more about actually using effectively our data. And there's another approach to design using in silico approaches to design screening sets and do virtual screens, which is building a biological network. So it's combining the idea of systems biology. So you could think about, for example, a target that you want to inhibit, and it's very difficult to drug these targets. For example, MCL1 or protein-protein interaction, which is really hard to inhibit with low molecular weight compounds. So what you could do is take one step back and look into the cell. What do you actually know about the biology of your target inside the cell? Maybe there's another protein that inhibits your target or phosphorylates your target or activates your target. And what you could do is actually build a biological network around your target and ask, can you actually activate a protein that inhibits your target? So it will be, compounds like that will be inactive in a biochemical assay, but they will be active in a cellular assay, which is what you care eventually. Eventually, compounds go into animals, in human. So you want to see the cellular activity. So you could actually have a desirable phenotype, for example, killing cancer cells and so on, not by directly inhibiting the target that is of interest, but inhibiting a different target which is upstream, but it's relevant to the target that you do want to inhibit. So it's indirect modulation of the target. So this is my view on silico drug discovery.
That is not what I expected to hear from someone whose expertise is ostensibly labeled as computational chemistry. But yeah, all that is very rich, and it very much ties into the course. So I mean, any of that is phenomenal. So what do you say we go ahead and try to get started?
Okay.
I'm going to start. I might mess up here and there. When I do, we'll just reset, and I'll say, crap, I screwed up, and then we'll reset, and I'll ask the question again, and I can roll into your answer. Right now, the system auto directs itself. So when you're speaking, you are on the screen, and when I'm speaking, I'm on the screen. And so what's on my screen is actually what the video that's being captured for the interview. So if you're talking and I look like I'm down writing or something, I am paying attention to you. I'm just maybe making notes for the next question or keeping track of things. But you just keep on trucking.
I have a meeting at 2:30. So is it enough if we have 20 minutes, or we need...
That's plenty. We'll be just fine. We'll go ahead and get started right now. Okay, I'll take a deep breath, and here we'll go ahead and get started. It's my pleasure to be speaking today with Meir Glick, and Meir Glick is a scientist at Novartis. And my first question to Meir is, Meir, could you describe your position at Novartis?
So I'm leading the in silico lead discovery group at Novartis in Cambridge. The group contains 9 PhD level scientists, and we're tasked with discovery of compounds that will eventually become leads and drugs, and we apply in silico or computational approaches to do so.
So, Meir, as much as you can describe, what was your career path that led you to your current position with Novartis?
So I studied pharmacy as an undergraduate, and I worked as a pharmacist for a while, and I figured out this is not what I want to be. And then I moved on and did my PhD in computational chemistry, mostly around predicting structures of proteins. Then I did my postdoc also in computational chemistry around the distributed computing, that is using the time that your computer is idle and to run virtual screening approaches using this technology, using hundreds of thousands of computers. 10 years ago, Novartis approached me and recruited me as what we call a lab head, which is a PhD level scientist here. 5 years ago, I took over the group, and since then, I'm the head of the group.
Fantastic. Now, Meir, we've been having some conversations before this interview started, and you repeatedly used the terms structure and ligand-based screening. So I'll ask you to explain a little bit what is the difference between a structure-based screen and a ligand-based screen when you do a computational approach.
So in the structure-based approach, we utilize the three-dimensional structure of a protein. This can come from example from X-ray. So we know what are the coordinates of the atoms in the protein, and we know where is the binding site of the protein. And we can have an hypothesis where should the compound and how should it bind. When it comes to ligand-based approaches, we actually ignore the structure of the protein completely. And what we utilize are compounds with known bioactivity that inhibit our target. So we use prior knowledge around the targets that we have, which are known inhibitors. So
when you do a virtual screen, how many molecules would you typically screen against your molecule, your target?
So we're talking about millions or even tens of millions of compounds. Although this is only a fraction of the chemical space, which is in the order of 10 to the power of 60 or even more, there's the practical element that when it comes to lead discovery, we usually propose thousands of compounds, so you want to screen compounds which are actually available. So there's no point many times to run a virtual screens on chemical matter that needs to be synthesized, which is a cumbersome process. So you want to exhaust all the chemical matter which is available in the archive, and in certain situations we go ahead and purchase compounds from outside, but normally we're talking about millions of compounds.
Okay. What kind of software package do you use to do this screening? Is this a commercial package that anyone would have access to? Is this something that's developed in Novartis, or is it a commercial package that's been tweaked to somehow serve Novartis's needs?
So for the structure-based approaches, that is using docking, that is to fit a compound to the binding site of a protein. We use commercial software packages like the Schrödinger package or Gold, Glide. When it comes to ligand-based approaches, we use our in-house similarity approaches that we developed. It's called FIPOPS. It's a 3D similarity approach where we use the three-dimensional structure of a compound. And we also use commercial packages like Pipeline Pilot to run a similarity search.
Can you say more about the similarity search? Similarity with regard to what properties would you be exploring?
So there are a few tiers of similarity searching approaches. There's one-dimensional, there's 2D, and there's 3D. Sometimes people also talk about 4D. So with one-dimensional properties, you're looking into molecular weight, C-log P, polar surface area, and so on. So these approaches are not that effective in virtual screening. When it comes to 2D similarity searching approaches, you're looking into the topology of the compound. That is, what is the scaffold, what kind of substructure the compound contains. So you're really looking into what atoms exist, what are the bonds between the atoms, the substructures, and then you apply these kind of principles. There are many what we call 2D descriptors. We tend to favor extended connectivity fingerprints that Cytigic or Pipeline Pilot utilizes. This method is very effective. We also use 3D similarity searching, which is using the entire 3D shape of the molecule, or think about the pharmacophore searching of the molecule.
Fantastic. So when a compound shows up as a hit in a computational screen, what are the steps that are involved in validating that activity in the computational screen before you would actually test the molecule in a chemical assay, a biochemical assay, or cell-based assay?
So I look into ways to contextualize the data. So for example, if I run a virtual screen against a certain kinase, I will take the virtual hits and I will ask myself, do they actually hit other kinases as well, if we're looking for more ATP competitive inhibitors? And if yes, then it gives me higher confidence that this approach worked well. So I always try to contextualize the data with any other experimental data that we have, and we have plenty of it. I also apply statistical approaches, for example, calculating p-value, to see that what is the probability that I found this virtual hit, not by chance. I look into the distribution of the hits. I look into the scaffolds that the virtual screening pulled. So what you want to have is also to provide eventually for wet biology a fairly diverse list of scaffolds or compounds to test. So there is no point of providing a virtual screen with 5,000 compounds with 2 scaffolds. So you want to cast your net as broadly as possible.
So when you find hits, can you talk about what would be your hit rate in a typical screen, if there is a typical range? I know that probably varies broadly, but what would be a typical hit rate? And then once you validate those activities as best you can, how many compounds would actually be screened in a biochemical test as a result of the initial in silico screen?
So the hit rate depends on the nature of the target. If it's a well-understood target, such as kinases, our compound collection is full of kinase inhibitors, and then therefore the hit rate is likely to be high. If it is the target is not well understood, for example, if you go after even a phosphatase, that we don't have enough chemical matter in the collection, I would expect a lower hit rate. It also, of course, depends on the screening technology and the assay type. So normally cellular assays tend to have higher hit rate than biochemical or biophysics assays, especially cellular assays that measure a decrease in the signal. Many times compound just stress the cell, and therefore will show up as false positives. generally speaking, what we call reporter gene assays have very high hit rate. It can be 5%. LCMS assays tend to have low hit rate. They have a low rate of false positives. Usually the hit rate is less than 1%.
So if you have a 1% hit rate, are you saying, let's say you screen 1 million compounds in a virtual screen, would you pass on, would 10,000 compounds be subsequently sent to a group to assay in a biochemical screen? Is that correct?
So yeah, usually assaying compounds is no longer a bottleneck. Mm-hmm. So if we have a solid in silico hypothesis, we could cherry pick, that is individually select 100,000 compounds, and we could assay all of them in a matter of less than a few weeks. So this is not a problem. The problem is understanding eventually the data that will come out of all these experiments. Do they show you the right biology? And many times they will go through a cascade of assays, biochemical assays, cellular assays, biophysics assays. And what you want to do is ask yourself, does actually the compound showing me the right biology, is this the right phenotype that I'm looking for? Does it really make sense? So I think what we struggle is actually understand our data. Generating it is fairly easy.
Excellent. So one thing we learn in class is that very often potency is related to affinity of the molecule for its target. And I believe that that's what many modeling packages try to simulate. What is the affinity between the molecule and its target? But then we learn about other approaches, things like drug target residence time, much more subtle ways to measure activity. Computationally, can computational approaches approximate these methods, or are these really the future of computational chemistry? Can you comment about these sorts of directions?
So when it comes to our business, you could think about lead discovery and lead optimization. When it comes to lead discovery, you worry less about the potency, and what you want to do is classify your collection into what's likely to be active and what's likely to be inactive. And you put these compounds through experiments, and you get the potency back in a more reliable and rapid fashion. When it comes to lead optimization, that is after 2 or 3 years that we have few interesting chemotypes, and what we're thinking is what is really the next compound that the chemist should make. And then this is a more cumbersome process because it's no longer taking existing compounds from the archive. You really need to make new compounds, which is a timely and an expensive process. Then you could apply more accurate methods that will hopefully try to assess the potency, for example, of the compounds. Based on my opinion, we're still not there yet. We still rely on fairly crude approximations of what we call non-bonding interactions. So factors like entropy are not well considered in these calculations. It's the future.
So computational chemistry, you are really primarily involved in the lead discovery stage, not the lead optimization stage.
I'm exclusively involved in the lead discovery.
Okay. Fantastic. Well, Meir, I want to thank you for giving some of your time to be interviewed for this course. And this is a topic I know very little about. So it's fascinating to hear your views and to shape what I've learned as well about this topic. So thank you very much.
No, thank you. Enjoy the conversation.
Great. Okay, Meir, we are